So on behalf of your advisor from Japan.
Okay. Okay, good morning, everyone. So happy that we're all here together, both virtually and in person there in Hilo. Just a couple of things for the people on Zoom. Remember to keep your microphone muted until it's time at the end where there'll be time available to ask questions. You can also ask questions by putting them in the chat and someone can read them out. And then if you're in the room after the presentation, you want to ask questions, please say your question loudly enough so we can hear, and maybe Anu can repeat it for the audience on Zoom. We're very pleased today to introduce Casey Anuhea Robbins. Anu has her roots in Waianae, Oahu, and at the beginning of her talk, she'll tell you a bit more about the places that raised her. As Anu explained, she finds the best way to give back to her community is through her passions. And thankfully for us, those passions include getting dirty and learning more about Hawaii's forests. Anu started in TCBES in fall 2020, and she did her undergrad at Washington State University. Because Anu first came to UH Hilo as a pipes intern to work on the Liko Napolina project as part of our team. I was thrilled when she wanted to enter graduate school and continue on this special restoration project. Anu has consistently shown an incredible work ethic, pushing through all the challenges of COVID, as well as sorting hundreds and hundreds of samples, and you're going to see the fruition of that today. She's also helped tremendously on the data management of the larger project, and we wouldn't be where we are today without her patience, her diligence, her carefulness. Anu not only did all the hard work of data collection and analysis, but I've seen enormous growth in her problem solving skills and her synthesis skills and communication skills. So it's a culmination of all those skills that you'll see in this talk today, as well as their passion and their core values. So just a preview, stay tuned for the finale, where she'll tell you all the lessons she's learned along the way in this journey. So we're so happy to have you, whether you're here in person or virtually, as she shares her story with you. So Anu, take it away.
Hey, aloha, aloha. Thank you guys for being here again. My name is Anuhea Robbins, and today I'll be sharing my research project and the culmination of what I've been working on for the past 3 years, with guidance and support from my committee members, Rebecca Ostertag, Robert Pett, Tracy Wagner, and Jonathan Koch. So yeah, this isn't just a defense or a thesis presentation. This is a story, a ka'au. And like every story, there's chapters. And in this particular story, there's 4. The hua, pa'alele, huaka'i, and huina. And the hua is a catalyst. And here I'll give I'll share about the events leading up to this project. For ha'alele, that's the preparation. Here I'll give some background context into the project as well as research questions. Huaka'i is the journey and encompasses the meat of the thesis, the methods, results, conclusions. And then last chapter is huina, the reintegration. And here I'll give some recommendations to restoration managers based on the results of this project, as well as some lessons I've learned along the way. Excuse my poorly drawn kuopa, but the theme I chose for this project is O ke kahua mamua mahope ke kūkulu, which translated means first the foundation, then the building. It emphasizes the importance of learning and making observations before you make decisions. And in my life and in this project, I have many kahua or support systems that have motivated me, inspired me to continue on with this project. The places that raised me are my kuhua. O kamaile unu lawa o ka'ala ku'umaumona, o kula ila lawa o poka'i ku'umau kaipili, Ke kupa au I ka moko koe anai he hale mola le I ka lā. These are the mountains and bodies of water that have raised me and have shaped who I am today. The people around me are my kahua, my community members, my ohana, my friends, my families, my peers. They really inspire me to be better and to learn. And they're just good fun to hang around with and make life worthwhile. And the best way I can give back to these places and communities is through my passions, which is aloha aina. You know, taking care of the land, like Becky was saying, getting dirty and just learning about different ways to understand these places and to help make them better in the future. And in 2019, my passions led me to an internship opportunity with the Pacific Internships for Exploring Sciences program, also known as PIPES. It's an awesome program geared towards providing undergraduate students with STEM scholarship opportunities in the Pacific. And PIPES partnered me with the Likonapalina Hybrid Ecosystems Project, or short Likonapalina, where I did a research project looking at arthropod communities among plant species. And that PIPES project led to this project. In restoration, it is often assumed that if we build it, they will come. This is based off the Field of Dreams restoration hypothesis, which states that, or is the idea that by increasing plant diversity, it'll lead to the return of wildlife. But a better question is not will they come, but who will come? It is unclear how plant communities used in restoration impact faunal communities and the factors governing these relationships. And this is especially true for litter-dwelling arthropods. Litter-dwelling arthropods amass a range of ecological functions and are important in driving key ecological processes and functions such as decomposition, nutrient cycling, and soil development. And thus, different arthropod assemblages can translate to differences in these ecological processes on both a micro and macro scale. And also, which is influenced by leaf litter composition. Leaf litter plays an important ecological role in ecosystems because they control soil light exposure. They make up most of the nutrient composition of soils, as well as provide food and shelter for fauna. So essentially, different leaf litters or plant species can translate to differences in arthropod community assemblages, which then can lead to differences in ecosystem processes and functions. And depending on what your desired restoration goals are, these differences in assemblages can play a huge role in the effectiveness of restoration strategies. However, the relationship between leaf litter and litter-dwelling arthropods is often overlooked in restoration. By understanding how different plant species and plant communities, restoration managers can make better informed restoration strategies. And a good way to study this relationship is through the use of functional traits. The functional or trait-based ecology is structured around the functional trait theory, which is the idea that the characteristics of an organism reflect its life history and individual performance. These characteristics, or functional traits, are described as the phenotypic characteristics that affect organism fitness and performance and are measured on an individual level. So for example, like leaf thickness or leaf height. While traditional ecology primarily focuses on using functional and taxonomic groups to describe these different, or to compare these different organisms, the use of functional traits is beneficial because it gives ecologists the ability to narrow in on the specific characteristics that underlie these species interactions. Numerous studies have looked at the relationship between leaf traits and arthropod community composition, but rarely do they focus on leaf traits and arthropod traits. The inclusion of arthropod traits can help us answer the question, if we build it, who will come? Or in the context of resource consumer relationships, if we build it, who stay coming for dinner? Yeah, so will it be the orbatid mite who prefers fungi to feed on? Or will it be a millipede who likes to eat dead organic matter? Or will it be an ant who likes, in this particular ant likes to prey on columbulas? So really incorporating arthropod traits can give us a better understanding of the specific trait associations underlying these arthropod assemblage patterns. In addition, most of these studies looking at this relationship are conducted in an undisturbed system, and rarely do they focus on disturbed or altered systems. An excellent place to examine these relationships is in the geologically young ecosystems of the Hawaiian Islands, where there are systems that are heavily degraded, such as lowland forests. Also, the Likunoplina project also presents a good opportunity to study this relationship in a very unique restoration setting. The Likunoplina project commenced in 2014 to test the efficacy of using different plant community assemblages in restoring a Hawaiian lowland wet forest. Setting a mood. Really.
Kind of where Sharon is.
Okay, thanks, Sam. Yeah, from a pool of 20 different plant species, both native and introduced, these plant community assemblages were created with 10 of these tree species based on two different factors. One, carbon turnover rate, whether these plants have slow or moderate carbon turnover rate, and two, functional trait profile, whether these plants have complementary or redundant traits. The first of which is what kind of comes off of the fast, slow leaf economic spectrum, which describes how plant species have different strategies or ways to survive to deal with their environment. And these strategies are characterized on a spectrum from fast to slow based on the way these plant species acquire and process resources. For instance, fast species exhibit fast nutrient acquisition, turnover rates, and growth rates, which are reflected in fast traits such as high nutrient content or shorter lifespans. Invasive plant species, for example, are considered fast, which allows them to outcompete slower plant species like native plants. And so in short, fast traits are associated with faster rates of processing, and slow traits are associated with slow rates. The Likunoplina project capitalized on this concept and used it to form 4 different plant community assemblages: slow complementary, slow redundant, moderate complementary, and moderate redundant. The study found that plant traits do in fact influence mite community composition. However, the project has yet to look into whether these traits influence, or how these traits influence the entire arthropod community, as well as the relationship between leaf traits and arthropod traits. So to fill this knowledge gap, I investigated the relationship between the traits of 20 different plant species and their associated arthropod communities to understand the influences of plant communities and plant species. The study is split into 3 different parts. The first part is looking at if plant species are differentiated based on the composition and trait values of their associated arthropod communities. Here, I characterize the arthropod community composition and trait composition of each of the plant species and compare them to identify assemblage patterns. So in this part, I expect to see how these plant species differ on a trait space, such as this, and to also identify key traits that are driving these differences in variation. I used the 20 plant species from the Likunoplina project, 9 of which are native, like Mamaki Lolu, and 11 of which are introduced, or non-native, non-invasive, like ulu, coconut, and mango. The second part looked at if there are correlations between the leaf and arthropod traits. This was done to determine if differences in arthropod community composition could be explained by traits. I expect there to be significant correlations between leaf and arthropods, which can help me to really narrow in on what traits drive these community assemblages. And then last, part 3. Here I looked at comparing the arthropod trait variation among the different plant species, the plant communities or treatments. This was done to determine how leaf and arthropod associations influence arthropod assemblages patterns on an ecosystem level. It also tested the resource specialization hypothesis, which states that an increase in resource diversity will also increase niche availability. In theory, plant communities with greater functional diversity should be able to support more functionally diverse arthropod communities. We tested if this hypothesis could be observed in this study within the complementary and redundant treatments. Sorry, kind of came on late, but yeah, you get the gist. So the leaf litter traits used in this study are leaf to petal ratio, leaf thickness, leaf mass per area, leaf area. Yeah, the traits are leaf thickness, leaf to petal ratio, leaf mass per area, foliar water content, foliar nitrogen, carbon, and phosphorus content, carbon to nitrogen ratio, and it's kind of missing in the bottom, but litter decomposition rate. And then the arthropod traits I'm looking at are body size, nitrogen 15 and carbon 13 isotopic signatures, nitrogen and carbon content, and biting force. And the reason why these traits were chosen for this analysis is because they are very much strongly associated with resource-consumer relationships. Yeah. The study site is located within the Kahua military reservation in Hilo, Hawaii, and is considered a Hawaiian lowland wet forest. Hawaiian lowland wet forests are usually characterized by elevations under 800 meters and annual rainfalls less than 2.5 meters. Anthropogenic and climate-induced disturbances have encouraged the invasion and colonization of introduced plant species in historically native dominant lowland wet forests, altering their biotic and abiotic composition. As of today, the forest is predominantly covered with introduced species, with native composition composed primarily of ohia, lama, and kopiko. The site is relatively isolated and protected from predators using electric fences, and the site substrate consisted of basaltic lava from the Kilauea and Mauna Loa volcanoes, formed about 700 to 1500 years ago. And as a result, the site has little to no soil development. So nutrient release is contributed solely by litter fall, fauna deposits, and basalt weathering. 2 arthropod sampling methods were used in this study. One to characterize the species arthropod compositions for each plant species, and then one used to categorize the arthropod communities of the treatment litter bags. So the first method, the species litter bags used a mix or single species litter in their litter bags. In comparison, treatment litter bags used a mix of species different litter based on the plant composition of each treatment type and their relative litter fall or relative proportion of litter fall. For the species litter bag, litter bags were deployed at the same time and collected at different time points. For species litter bags, that was 1 and 6 months after deployment. But in this study, we only used time point 2 because the samples from time point 1 were compromised. And then for treatment bags, that was 1, 4, and 6 months after deployment. And then once we had the litter bags, arthropods were extracted using Berlizzi funnels and then sorted and identified by proxy taxa or morphospecies, and then later identified to the lowest taxonomic rank. Average leaf litter trait values for each of the plant species were obtained from the Restoring Ecosystem Services 2, or REST, which is pretty much has a database of a lot of plant functional traits. Average litter decomposition rate was calculated using a first order negative exponential decay equation. Stable isotope ratios, nitrogen 15 and carbon 13, and estimated nitrogen and carbon content were measured on 35 different morphospecies, 67%, which represented 67% of the total litter bag abundance. Specimens were dried at 70 degrees Celsius and for at least 48 hours, and then folded into tinfoil capsules and analyzed using an IRMS or isotope ratio mass spectrometer at Hilo Analytical Laboratory. Dry body mass was used as a proxy for body size and was measured on 63 morphospecies, representing about 90% of the total litter bag abundance. Specimens were dried at 70 degrees Celsius for at least 24 hours, like the isotope, like the stable isotope specimens, and then weighed and averaged. Arthropod biting force was measured on 8 orbatid mites. Orbatid mites were chosen for analysis because they are by far the most abundant taxon, comprising about 50% of the total litter bag abundance, and because their similar mouthpart structures were ideal candidates for comparing biting force. Biting force is defined as the biomechanical force in newtons exerted by the tips of the mouthpart, and in the context of orbatid mites, that's their cheliceraries. So the equation is Fn, which is head volume or abductor muscle volume. And then Fn is abductor muscles, which was, sorry, this gets kind of complicated. So Fn is the head volume or abductor muscle volume force. And that was using the head volume with the assumption that abductor muscles usually comprise most of the head volume. And then Ln is the width of the mouthpart structure. And then L out is the length of that mouthpart structure. But it was hard for me to measure the width and lengths of these mouthpart structures without dissecting them. So I had to estimate them using the rotella, which is that tooth appendage in green there. And just a side note, the rotella is used to hold down prairie wild they eat. But yeah, so I estimated those lengths using the rotella. So to measure the different mouthpart structures of these arthropods, I measured them using images derived from a scanning electron microscope, or SEM. For SEM biological preparation, I first had to dehydrate the specimens in an ethanol graded series, and then dried them using a critical point dryer, and then coated them in gold using sputter coater, and then analyzed them in the SEM and measured them using an image measurement software. Community mean trait values were used to characterize the arthropod trait composition of each plant species. So to calculate the community weighted means for each plant species, for each trait, for each morphospecies, the average arthropod trait values were weighed by the relative abundance of each of those species to calculate these community weight mean values. For part 1, I analyzed the plant species were differentiated based on the composition and trait values of their arthropod communities. This study found that plant species do, in fact, facilitate different arthropod assemblages and thus different arthropod trait values. I ran ANOVAs or Kūkulu if the data were not normally distributed to test for differences in mean arthropod total abundance and species richness among the plant species. While the study found no difference in total arthropod abundance, plant species did differ in species richness. In particular, monkeypod, which had significantly lower species richness than most of the other species. especially so for Mamaki and Lolu. I further analyzed differences in arthropod abundance by taxa among the plant species using a series of Poisson regressions or negative binomial regressions for models that were overdispersed. Introduced plants were found to have greater Formicidae and Diptera abundance, or ant and fly abundance, which could be alluded to the fact that these most introduced plant species in the study have larger, fleshier fruits. So it kind of makes sense that these ants and flies would be attracted to these fruits or plants with fruits because of the food that they provide and the habitats they provide for, say, Diptera egg laying. And also, coconut was found to have the greatest Formicidae abundance out of all the species, all the plant species. Much was composed of the ghost ant, the Tapenoma species, which is well established here in Hawaii. This makes sense because ants are associated with palms because the crevices between their fronds act as good nesting locations. I further analyzed differences in arthropod abundance by trophic yield. Trophic yield are functional groups based on the arthropod's diet composition. So for example, fungivores, herbivores, and predators. This particular study found that Hilo and Anuhea had significantly higher detritivore abundance. For Anuhea, this could be due because an abundance of dead wood caused by Anuhea dieback, which is a natural phenomenon, straticitis, wilt, and canker could lead to more dead wood, which equals more resources, food, and habitat for detritivores. As for Hilo, it's a little bit harder to understand why we see these observed results, just because I haven't been able to find a lot of studies looking at Hilo, particularly with arthropods. But to give it some context, Hilo had the highest total abundance, which, increases the likelihood of having more detritivores, just based on having more arthropods in general. The study also found that Mamaki and Lolu had higher fungivore abundance, which could be because they facilitate good habitat for fungi, their food resource. For Lolu, Lolu had comparably higher leaf area, which could in turn translate to more shade, which in turn translates to more fungal growth and more resources for fungivores. For Mamaki, you could be seeing a high fungivore abundance because of Mamaki rust, which is a fungal disease. Ambrosia beetles, which are fungivores, have been shown to prefer Mamaki as host species and can act as vectors in the spread of fungi, increasing fungi populations. To identify the key traits responsible for trait variation among the plant species, I used 3 principal component analyses, one looking at just leaf-related traits, another looking at the community-weighted mean arthropod traits, and then a third looking at the combined leaf and arthropod traits. And the PCAs show that plants are differentiated by their leaf-related traits, most of which was influenced by leaf palatability and quality. So PC1, the x-axis, showed that, or PC1 was positively associated with carbon to nitrogen ratio, leaf mass per area, and leaf thickness. Whereas PC2, the y-axis, was negatively associated with litter decomposition rate, leaf area, and leaf to petiole ratio. This shows that plants are mostly differentiated based on their leaf palatability and quality traits. The PCA looking at the community-weighted mean arthropod trait values among the plant species explains 78% of the variation among the plant species, much of which was influenced by carbon and nitrogen nutrient content. Arthropod PC1, the x-axis, was positively associated with carbon and nitrogen content and binding force, and negatively associated with carbon-13 and nitrogen-15 signatures. Whereas PC2 was positively associated with arthropod body size, nitrogen-15, and nitrogen content. Which is, so basically what this PCA is telling you is that PC1 differentiates arthropods composition based on nutrient content and negatively associated with trophic level, and PC2 is positively associated with larger, more predatory arthropods. Plant species are differentiated by their leaf and arthropod traits. This PCA shows that PC1 is positively correlated with leaf nitrogen content and phosphorus content, and negatively associated with leaf carbon to nitrogen ratio and leaf mass area, whereas PC2 was mostly influenced by, or had a negative association with arthropod nitrogen content and carbon content, and positively associated with carbon-13 signatures. So pretty much in summary, PC1, the variation in PC1 is driven mostly by leaf traits, whereas PC2 is mostly driven by arthropod traits. To compare the arthropod community composition of the plant species, I ran in a permanova and visualized the results using an NMDS plot, or non-metric dimensional scaling plot. Plant species were found to significantly have significantly different arthropod community compositions. There are several interesting observations that we can make from this plot. One, that introduced plant species show more within group variation than native plant species, suggesting that introduced plant species have a more wide range of arthropod community compositions. However, there does not seem to be any distinct differences between introduced and native plant species, except for monkey pod, which is in that little corner back there. It's important to note that monkey pod is the only nitrogen fixer in this study, and it also was found to have the lowest species richness out of all the plant species. So what's this telling me is that honestly, I don't have all the information here. And so it might be important to include other plant traits, such as root-related traits, to fully understand why we see these observed differences. Another interesting observation is that ulu and hala have very similar arthropod community composition, which is denoted by that clustering. But it's really interesting because in the previous PCAs, ulu and hala didn't have similar leaf or arthropod composition. So this really does hit the fact that I don't have all the information here to explain why we see these variations or similarities. And that maybe including more traits can help me to better understand these relationships. For part 2, I looked at whether there are correlations between leaf and arthropod traits among the plant species. Leaf and arthropod traits for the plant species. This was done to determine if differences in arthropod composition could be explained by trait associations. I ran a series of Pearson correlation tests or Spearman rank tests if data were not normally distributed between leaf and arthropod traits, as well as the PC1 and PC2 scores of the leaf trait PCA and the arthropod trait PCA from part 1. While significant correlations were found between leaf and arthropod traits, the correlation or the relationships were better summarized in the correlations between the PCAs. The study found that there was a strong negative correlation between leaf PC1 and arthropod PC2. And if you remember back to part 1, leaf PC1 is negatively associated with leaf thickness, leaf mass area, carbon to nitrogen ratio, whereas arthropod PC2 is positively correlated with body size, arthropod nitrogen 15, and arthropod nitrogen content. So essentially, this implies that acquisitive plants with smaller, thinner leaves and faster nutrient release are strongly associated with smaller arthropods lower in the tropic position. The correlations also found that a strong negative correlation between leaf PC2 and arthropod PC1. In part 1, leaf PC2 was found to be negatively associated with liver decomposition rate, leaf area, and leaf to petio ratio, whereas arthropod PC1 was positively correlated with carbon and nitrogen content and carbon-13, and negatively associated with carbon-13 and nitrogen-15. And this correlation implies that plant species with slower conservative traits, like large leaves, thick leaves that decompose slowly, are more associated with larger, more predatory arthropods. So in short, part 2 found that arthropods of lower trophic levels were maybe strongly associated with fast traits or fast plants, because we might see this because fast plants have more palatable and nutritious leaves as compared to conservative plants with thicker, harder to chew or bite and process leaves. And this kind of reflects their resource-consumer relationship. However, arthropods of higher trophic levels, like predators, don't exhibit this resource-consumer relationship as much as some of the arthropods lower in the tropic position do. And so they could be associated with these slow conservative plants for other reasons, such as slow plants have larger, thicker leaves, which could provide more shade, shelter, and protection from predators, as well as provide a more preferable microclimate. This shows that leaf and arthropod traits can be used to understand arthropod assemblage patterns. For part 3, I compared the arthropod trait variation between the different plant communities or treatments to test whether the resource specialization hypothesis was observed in the study. And in short, it was not. No differences were found in arthropod trait variation between complementary and redundant treatments, but we did find differences between treatment types. For instance, body size variation was the highest for the slow redundant treatment, the most conservative treatment type. And this could be because of multiple reasons. It could be because of their plant community composition. A lot of the plant species in this treatment have both the highest and the lowest community weighted mean body size values. Plants in this community also have high leaf thickness, leaf mass per area, and carbon to nitrogen ratio, which is pretty much slow plants. And so slow plant species essentially are more correlated with larger, more predatory arthropods. And then high body size values can be attributed solely to mountain apple and copico. So part 3 found that there are no difference in arthropod trait variation between complementary and redundant treatments, which fails to support the resource specialization hypothesis. But body size variation was greatest in slow redundant treatment. Leaf traits were shown to be related to arthropod traits of plant species, and this relationship can be observed on a plant community level. So some recommendations I can make, or I can give restoration managers based on this project. One is that plants do matter. The plants that you pick in your restoration strategies can have a great impact on ecosystem processes and functions, which can in turn determine the success and effectiveness of these restoration strategies. 2, restoration managers, like I said earlier, should select plant species based on their functional traits to meet specific restoration goals. And this is because plant traits have been shown to be associated with arthropod assemblage patterns. And essentially, fast plants equal faster ecosystem processing rates and traits. Slow plants have slow processing rates and traits. And then the last recommendation I can give to restoration managers is to consider using introduced plants in your restoration strategies. Introduced plant species have been shown to facilitate similar arthropod species richness and abundance as native plants, as well as having greater variability in arthropod community composition than native plants. They can contribute a lot of very, very important ecosystem services that native plants fail to do, and can help aid in meeting specific restoration goals. So the studies show that establishing significant linkages between traits of leaf litter and litter-dwelling arthropods can deepen our understanding of the principles governing these relationships and guide restoration managers in creating strategies that are inclusive to species interactions on a micro scale. Like every story, there are lessons learned, and here are some of the lessons I learned throughout this project. Lesson number 1. Be okay with NA values. And NA means not applicable or not available. When I'd first written up the proposal for this project, I'd been pretty set on measuring each trait measuring every trait for each morphospecies. But when you have like 200 plus morphospecies, it's kind of hard to measure them all. So yeah, you kind of have to be okay with what you can do and what you can't do. And because research is a Kūkulu thing, making space for others to fill in these gaps is really important as well. Lesson number 2, give every sample its own name. Just like how names are given to people, places, and things to give them purpose and value, you should do the same with your samples. For our study, we were naming them by quadrant. We were naming each sample by quadrant and plot. And because it's randomized, sometimes we'd have the same plot and quad. So some of the samples have their similar names. So that made it really hard for us to distinguish between each. So lesson learned, give them their own name. And then lesson number 3, find your Kahua or support system. Without my Kahua, this project would not have been possible. And so yeah, I'd like to acknowledge them. First, I'd like to acknowledge my committee members, Dr. Rebecca Ostertag, Robert Peck, Dr. Tracy Wagner, and Dr. Jonathan Koch for guiding and supporting me throughout this journey. Thank you to the project funders, CERDEP, National Science Foundation, Hawaiʻi Army National Guard Environmental Office. Big shout out to the PICE office who are here today. PICE Ohana. Thank you for really being the catalyst of this project and inspiring other undergraduate students like me to pursue STEM careers. Thank you to my graduate program funders, Kamehameha Schools and Hawaiʻi Veterans Memorial Graduate Scholarship for funding the entirety of my program. Thank you to the UH Hilo Analytical Lab and technicians Eric Johnson and Tara Halitsky for helping me to run my samples as well as teaching me how to use the RMS. And then also a shout out to UH Hilo STEM Laboratory, in particular, Dr. Cheryl Jansecki for teaching me how to use the SEM. And then last but not least, thank you to the Likuno Plena crew for literally doing all the heavy load, litter bag creation, litter bag collection, arthropod sorting. So yeah, this project would not have been made possible without them. And yeah, that concludes my talk. Pipi Holoka'o. So yeah, thank you guys for being here and sharing your space with me and giving me the opportunity to share what I've been working on with you guys. I was really excited, also nervous to do it, but I'm glad I did it. And it was nice to see some faces I haven't seen in a long time. But yeah, I would like to open Zoom or in person for any questions that you guys may have for me. And if now is not a good time to ask questions, you guys can always email me. I didn't add it here, but it's crobin6@hawaii.edu. So hit me up if you have questions. Yeah, thank you guys.
There's something in chat. Yeah.
Oh, thanks, Mom. I mean, it's good if you guys don't have questions, because that's the part I was the most worried about.
What's your favorite bug?
The Columblas, I featured them in the beginning. Columblas or springtails. Anyway, they're like. Anyway, Columblas, springtails are my favorite, just because they look really cute. They have different colors. Some of them are like, no, they have like my favorite color palette, like pale blues and purples and pinks and whites. So they're my favorite. They're the hardest to sort though, because they're so fragile and they just float at the top of the alcohol. So you're just like swimming around with your forceps, kind of grabbing. But yeah.
This might be really simplistic for the analysis that you did. It was a lot more advanced than I'm thinking, but you divided the plants into introduced and native species. And do you think that you could learn anything from doing the same thing with the arthropod community and looking at their associations
Oh,
there?
yeah. So Stephanie asked.
Can you repeat the question for the Zoom people?
Yeah, can do. Stephanie asked if, so she said that I looked at comparing native and introduced plant species and that whether I can do the same with arthropods, native and introduced arthropods. And that's a really great question because one of the hardest things that I came across this study was that there's not a lot of literature or a lot of knowledge on whether arthropods here are native or introduced. And it's particularly so for the soil arthropods. So a lot of it, we don't know. We don't know whether something is native or introduced for a lot of these species. And some of them haven't even been identified yet. So that did present a lot of major issues in my study. But, you know, studies like this that are trying to focus more on the soil litter arthropods are important so that we can, you know, increase our knowledge base on this. So yeah, hopefully, I'm sure there's other research going on that's looking at what do you call that? Naming and identifying these arthropods. But yeah, I think we have a lot more to learn about them. Yeah, thank you for the great question. But yeah, unless anybody has other questions.
Yeah, no, I have a question for you.
Go ahead, Bob.
Yeah, first of all, that was outstanding. You really covered a ton of material very well in a pretty short period of time. I have a question. So you weren't, for your species litter bags, you were not able to use the first time point for understandable reasons. But I'm just wondering how you think the litter may have differed between those two time points, and if the arthropod communities may have been expected to differ as well.
Yeah, good question, Bob. So the differences with the time point is that the reason why we did different time points was to capture the temporal differences between these arthropod communities. And so the 1 month kind of just gives you a preliminary analysis of what's coming in super fast. And then with the 6 months, you know, the litter bags are set out longer. And so they have a lot more time for resources like fungi and bacteria to accumulate. So you kind of see more a developed arthropod community composition for these leaf litter. So essentially, with time, it kind of worked out because with time point 1, I didn't expect to see much of the arthropod community assemblages explained, whereas the 6 months you have time for arthropods to colonize these leaf litter. And so you kind of get more of a representation of the natural arthropod assemblage patterns for these plant species. Does it kind of hit your question? So I don't expect there to be, I do expect there to be differences, but I do think that time point 2, which we used in the study, was better representative of these plants or these arthropod assemblages for these plant communities, plant species, sorry.
Yeah, no, I think that's a good answer. You know, and you can think of it as I mean, it's a succession, right? So the community goes through a natural succession, like with pioneering, you know, early on, you get maybe pioneering species, and then towards the end, as the vegetation the leaves are more decomposed, as you say, you have more advanced community or more fully developed. So yeah, just curious if you would have had all of your analysis during the first time point and then all of your analysis during the second time point, and those would have been completely separate. It's possible you could have had different results.
Yeah, I do think if we did include the time point or 10.1, there would be more samples, so it would be able to better explain some of the differences. But yeah, that was just not possible, so.
Yeah. Thanks.
All right.
I had one question for you, Anu, as well. So great presentation. A question for you is, you know, when you started this project, did you have any expectations or predictions as to what you were going to find in your project relative to, you know, the different tests you did? And, you know, were the results that you found, were they on par with your predictions, or was there anything interesting that surprised you, you know, as you were going through your project towards the end?
So in my PICE project, I looked at comparing arthropod community assemblages among different plant species, similar to this exam. But that study, I found no differences between arthropod species richness or abundance by taxa or stuff. So honestly, when I went into the study, I wasn't really expecting to find much differences. And it kind of was just a gamble. When I did the study, there's not a lot of like resources for me to use, especially since this is such a new or recently developed technique using both leaf and arthropod traits. So yes, I was expecting to find some differences and significant differences, but I just didn't know where. So I'm really happy with the results I got. I think they helped to explain a lot of stuff that I wasn't really expecting to see.
Thank you. And then actually I have one follow-up to that. If you could go through the process of this again, based on what you've learned, is there any particular trait that you think, well, you know, based on what you've observed, like, is there any particular trait that you could, like, you know, if you were to do a follow-up to this, you know, is there anything that really stuck out to you that you would maybe propose for a future study?
Yeah, so one of the traits that I was reading about that was shown to be significantly correlated with biting force is lignin, which is pretty much like the toughness of a leaf. But I wasn't able to measure it because I didn't have the resources or instruments that you use to really measure it. So that's something I had to leave out. But that's one trait that I would like to look at, leaf toughness, when it comes in. And then biting force was a really conundrum because initially I wanted to compare biting force between different taxa, but with the method I had a hard time comparing them because they just weren't comparable, because they have different mouthpart structures, and these mouthpart structures have different functions and stuff. So, like for example, the mites, they, you know, chew, bite, but then you also have stuff that siphon nutrients out of leaves and stuff like that. So that's one trait that I wish I was able to dive more in depth. And it's just it's a lot of work to even just do those 8 orbat and mites. So, but yeah, ideally I would like to look more at the biting force, because that really has been shown to be significantly correlated to the resources that they eat. So, yeah.
Thanks, Anu.
Thanks, John. But yeah, if there's any more questions, again, feel free to email me, crobin6@hawaii.edu, or yeah, just ask around. It's Hawaii. Yeah, thank you again for everybody for tuning in virtually and in person. I truly appreciate it to share what I've been working on, connect with these people. But yeah.
Thank you.